Hello and welcome to Behind the Episode, a rare disease advocacy podcast by Cure AHC, created by AHC parents and advocates for our amazing AHC community. I'm Izzy Duval, your host and the Vice President of Patient and Family Services for Cure AHC, a nonprofit organization committed to finding treatments and an eventual cure for alternating hemiplegia of childhood, or AHC, and other ATP1A3 diseases. Since we dropped the first episode, I've been so excited and honored to hear from so many families around the world, from UK to Argentina to Egypt, and of course here in the States. And on behalf of our podcast team, which is myself and Hannah and Corey Zimmer, we want to extend our gratitude to everyone listening and reaching out. As AHC parents ourselves who are often living in the trenches alongside you, these kinds of connections truly help us to stay motivated and moving forward. So thank you. This episode features the long-awaited discussion with Nina and Simon Frost of Rare Hope, formerly known as Hope for Annabelle. The Frosts are true trailblazers leading our community toward real therapies and real hope for a cure. Nina Frost is the driving force behind the international patient-led AHC research program, working side by side with families, scientists, and industry professionals to turn what matters most to our patients and our community into real life-changing therapies. And Simon Frost, of course, is the past president of Cure AHC and a sitting board member across, I believe, 7 different major rare disease organizations. Simon brings a bold systems-level vision using the same large-scale strategy that he built in the business and economy world to create faster, smarter pathways to treatments for our children and for rare disease communities everywhere. In this episode, we are talking about all things gene therapy and AHC, where the science stands as of right now, what the recent prime editing success in AHC mouse models actually means for us, and what comes next as we look toward the future. Months ago, we gathered your questions as a community, and this conversation is where those answers will live. Hannah Zimmer joins us today on this episode as co-host and community engagement lead, making sure that we answer all of your questions in a way that's understandable. This is a longer episode because we didn't want to cut out any of the information that feels so important and relevant and hopeful to our families, but we also know that time is very limited. So we've created chapters and timestamps that live in the show notes so you can skip around and watch the parts that are most important to you. Before we dive in, I want to just pause on something that honestly took my breath away when we recorded this discussion. The recent prime editing success in AHC mouse models is the first time a neurological or central nervous disorder has ever been genetically modified successfully in a living animal. Not a petri dish, not isolated cells, but in a living mammal. We've seen CRISPR breakthroughs in the eye, liver, heart, and blood, and you may have heard about baby KJ or the FDA-approved treatments for sickle cell disease and metabolic liver disorders, but crossing the blood-brain barrier and successfully editing a neurological disease in vivo has never happened before until now. That's so exciting. So let that sink in. And with that, let's dive in. Let's go behind the episode.
Annabelle Frost! Do you want to bring Annabelle with an extra L in here? You okay?
She has these episodes and they freeze her muscles.
Annabelle? Here, come look at Mama. Can you look at my eyes? Okay, can I see it up close?
Just looking at her, I can tell she's in a lot of pain.
It's a really tough disorder. Probably one of the most challenging that I've ever worked with. I think it is the most challenging disorder, and the reason is because of the uncertainty level.
Our life is completely structured around what works and what doesn't work for Annabelle. Every day she has to have a 2-hour nap. Wherever we are, everything else revolves around that.
We went swimming and you were the best little girl in the whole wide world.
She falls all the time. She eats, she chokes. She will go limp. She'll lean her head back. She loses her ability to talk. Some AHC kids have these episodes for days on end. They'll have it all day long, go to sleep. It'll resolve a little bit with sleeping. They wake up, they have maybe 10, 20 minutes to eat and drink, and then it'll start again.
It's more and more clear that it shortens the lifespan, and we are aware of children that have died suddenly. And as their neurologist, not knowing, is that child going to be okay today? Are they going to have a good day? Are they going to get through the day? Or are they going to have a life-threatening crisis situation?
We have been part of the AHC world for 5 years. And over that 5 years, we've been as active as we could be in terms of trying to organize the community, develop therapeutics. It has been our desire to make AHC an easy target for researchers, for pharma. How can we establish AHC as a disease that researchers want to work on, even if they're the best in their field, especially if they're the best in their field? Because you're a tiny little spot on a huge map of rare diseases.
When we started, it was with pursuing a single therapy. And the question was, can we adapt this form of therapy for AHC? What it is now is a platform of we're pursuing multiple therapies of different mechanism of action, all at different level of intervention. From the gene to the transcript to the protein, testing to see if the disease can be mitigated.
We need to develop this portfolio approach. We need to develop multiple different therapeutic strategies. We need to pursue them in parallel.
And that's why I want to wait at least until the end of that experiment.
Natalia and I approaching scientists, hey, we understand that this is your area of expertise. We want to develop a project here with you. It is a very different approach than we've used in the past.
You have these separate labs in different research institutions. They were aware of each other. They would come across each other's research, published research, or they would meet at conferences. But each lab is its own ship that has its own captain that decides where to swim, where to sail the ship. Nina has brought together the scientists and the medical doctors that are already interested either in the disease or this gene. But now this research is collaborative. It's synergistic because now they feed off each other and they make these points of connection.
Our driving mission is to help systematize the treatment of rare diseases, to establish a platform approach for rare diseases where rare diseases can kind of go through a similar process no matter what the disease is.
The most clinically relevant best thing that I would like to see is a reduction in spontaneous deaths, particularly in male mice. That's what I'm seeing.
Dramatic improvement.
Putting all of these pieces together, we are the hub of all of this energy and focus.
So I'm super excited today to welcome some of my favorite people to the podcast. We have Nina and Simon Frost here from Rare Hope, also a part of Cure AHC, where Simon was previously president and Nina is an honorary board member who we see all the time at meetings. And they run Rare Hope, which was formerly Hope for Annabelle. And so we're super excited to invite them into the podcast today to discuss the science, the research, the treatments that are upcoming for AHC patients, and kind of how all of that works. So welcome to the pod.
Thanks, Izzy. You're one of our favorite people too.
You are too, Hannah. You guys do amazing work.
Yes, definitely. Well, Nina, I want to start with you. Could you share with us the story of Annabelle getting diagnosed with AHC and how that affected your family, and then what led you to want to create Hope for Annabelle and get involved in the research? I know that both of you have a lot of educational background, but I do think it's been a little bit of a new road that you've traveled in terms of the genes and the science and everything. So I would love to just hear your story.
Well, sure. Annabelle was diagnosed kind of late with AHC. She was 2 years old. And she had her first episode on the delivery table. So within minutes of being born, she was having an episode. And we didn't notice it at the time. But when we went back and we looked at all of our video from, the instant that she was born and taken and, wrapped in blankets and everything, there she is in the delivery room having an episode. But we knew that that there was something that wasn't right. We thought that she was having seizures and we thought that she had epilepsy. And for 2 years, we didn't have a diagnosis beyond epilepsy. But for 2 years, we were also back and forth to the hospital in ambulances. She would have what we thought were seizures, and it would, start, and then she'd have another one, and then she'd have another one. It seemed like it was clusters of seizures that were increasingly scary and really just terrifying when she was that age. And it was 2 years of real uncertainty. And we finally did kind of stumble across the diagnosis, we also stumbled on the Human Time Bombs video with Suna in it, the beautiful film that Ziggy had made. And we saw Suna's suffering, and we saw so much that just seemed identical to what Annabelle was experiencing. And it was the most awful gut punch of a feeling, where we were looking at Annabelle's future, and we were looking at, this other beautiful girl who was clearly suffering from something that was just so awful, and yet here she was, this amazing kid, and we were just looking at Annabelle's future, and it was just such a moment. And at that point, within a few weeks of seeing that video, and we stumbled across that video before having her diagnosis, but it hit us so hard and was so familiar that we thought, hey, this is what it's going to be, and it was. And it was a very, very difficult time, and I think, Simon was such a hero in those days. He threw himself into, well, don't smile, it's true, my darling. You threw yourself into finding out everything that you possibly could, not just about the disease, but about the possible paths to a therapy. And it was from the very beginning, I think Simon kind of set the stage for us of, not only are we going to be very, very deep into the scientific detail around this disorder and around the possible paths to a treatment, but we are going to do something about it, and we're going to make a difference. And I think that has been the mindset that we've had from the beginning. And I say we from the beginning, but truly the first few months, I would have rather been in bed with a blanket over my head. Right? It was a very difficult time to kind of lose all of your expectations for what your future would look like with your family, and encounter something that was different and that seemed heartbreaking at the time.
Yeah, I think we can definitely all relate to that. I know I spent a good couple of years trying to not also lock myself into a dark room and hide and cry every nap time. So I understand for sure. So how did you then create Hope for Annabelle? I know Simon was doing all this research, he was digging into it. What made you think like there was a possibility to creating something that could help Annabelle and other children?
So Simon started researching. He found Natalia Morsch, who is, our dear friend at this point, former NIH scientist who we love dearly, and who's really just a genius, and one of the most kind of informed scientists who just lives and breathes AHC in the way that we do. And they were really making progress, and they really had this idea that pursuing a gene therapy for AHC would be kind of the highest likelihood of success, and the way that we could, impact the most patients the most quickly. And so, they were putting these pieces together, and we thought, well, let's start a way to rally our community, the people, the friends and family that we know who care about us, who care about Annabelle, and let's start raising some money. And so that was kind of the intent initially, was, inspired by Annabelle, but, looking to do something for all of AHC. And so we started raising money, and that was my role early on. We had a big event in 2019, which took a full year to organize. It was a big, fancy gala type event, similar to the one we had this year, but on kind of a less grand scale. But we raised almost $900,000 in one night, and that kind of launched things for us and put us in a position to be able to kind of execute on the science.
It's incredible. I just kind of want to take a pause and think about how what you all have done has paved the way for so many of us. I mean, Dallas was born in 2018, and so I can't help but think when you're talking about this is the year that we started Hope for Annabelle, this is the year we did this, in a sense, Dallas was just coming onto this earth, and you guys are already over there creating things that could potentially help her long term. And I don't know, Hannah, I'm sure you feel similarly. Do you have anything you want to say in terms of that?
No, I think we were the same way. We, as obviously everybody, I think most families see ticking time bombs, but for us, we being in the DC area, the Frost family was the first ones we found. And so Hope for Annabelle and their website and the videos of your family were the first ones that we came across. And I think kind of coupled with that, it was like, wow, look at what this family has already done. Look at what compared to when Annabelle was diagnosed, and the progress that you all have made. It truly was gave us hope when we were first diagnosed. So we're really grateful for your efforts leading up to that.
Yeah.
And I do want to say that we felt the same way when we got into this disease too. I mean, we definitely, to Nina's point, felt like we were on our own and we needed to figure this out. But when we did start to figure it out, there were so many kind of shoulders of giants that we could stand on. There were, a lot of very high quality papers about this. We had people like Shoyano who got his MD PhD at like 5 years old. I'm exaggerating. But there are just incredibly advanced people in this disease that we felt privileged to be able to work alongside or follow behind. And the same thing is true with you two. Like, you getting into this disease now, you're the first face or the first voice that people hear, and you're creating the same kind of continuity with this disease that hopefully kind of we contributed to.
What we've really tried to do is kind of build a hub or build a node where all of the pieces, that when we got to this world, there were lots of pieces in play. There were lots of things that were happening, but they weren't coordinated and they weren't centralized and they weren't focused on, the interests of patients. And I think that's been what we've really tried to do over the last 8 years. Is it 8 years? Oh my gosh. It's 7.5, 8 years now. But what we've tried to do is understand the patient experience, understand what the burden that they experience is, understand what their families and caregivers are, most concerned about. We try to understand all of that as context, and we try to take those patient experiences and really insert them into the research that we are hoping to advance. And I think that's been such a critical piece of us. Not only are we creating a place where different researchers can collaborate, a place where we can kind of assemble teams of really incredible people, but we can kind of put them in a position to work together in ways that might not have been possible before, or ways to assemble resources and think about gaps that, where there are knowledge gaps that we need to fill. We've tried to kind of put that system into place, put kind of an organization on all of the good things that were happening. But at the center of it, we've tried to put the patient experience. And we haven't done that by having our opinions on what we think are important. We've tried to really take data and transform that data into actionable kind of insight that we can use as we're thinking about where to invest our resources, the community's resources, and how we can best advance research to serve the needs of the patients that we serve.
Yeah. I think hearing from Simon about how this recent in vivo prime gene editing was the first of its kind for this type of disorder in the brain, and then realizing, in thinking back, to 8 or so years ago when you were talking to Natalia and thinking about what would be the best treatment option for our patients, it's actually just really kind of amazing to be sitting here with you all right now, realizing that this was, a thought you had in your minds, and really you've been trailblazers in many ways, and I already knew that prior to this conversation, but just seeing the road and the pathway to where we've gotten, and the fact that this brand new, never happened before research has occurred is really incredible. I don't know, with this recent project, how, I'm sure there's lots of feelings and it's complicated, but how did that feel, knowing that you were able to accomplish something that you've been working after for 8 or so years?
I mean, it's such an achievement, and it's such an incredibly strong team. I mean, someone like David Liu is an incredible person to work with. He's not only, an inventor and an innovator and a field leader, but he's, empathetic, and he cares about patients, and he's a wonderful person. And so working with teams of field leaders makes things possible, I think, for everyone across our disease community, whether it's David or whether it's the other scientists that we're working with. We are bringing together truly some of the just the most incredible scientists that are working in the world right now. And so there's a huge amount of excitement about all of these things that are happening, but it really is, I kind of am compartmentalizing the scientific excitement, which is massive, with the kind of thought about how this is just one more step on the path that we are all navigating, right? Like this is a really great success. It is actually opening doors across all kinds of other things. There are all kinds of other collaborations and partnerships and technologies that people are approaching us with because of this success. So it's opening a whole lot of doors that, that we would not have thought possible. Funding, grants, things like that as well. But at the end of the day, the end game is making patients' lives better. And so this is just, it's a moment to celebrate, and it is getting us a lot closer to that point. But I think we always have that context that, the end game is still the patients.
I'm going to kind of expand just a little bit on what Nina said. Like translating that to the next level, like making this available to a wider population than just AHC is so obvious with this particular team. I mean, Nina mentioned David Liu. He's the preeminent guy in gene editing at this point in the world. Tim Yu is the same with ASOs. He's one of the top people in the world. Jackson Laboratory, Kat Lutz and Marcus Terry are, again, field leaders. And the combination of these people, they're all working together now to develop therapies in the most practical, most production line type of way for as many patients and diseases as possible, led by AHC. So seeing that possibility of translation and seeing it actually happen is another exciting prospect.
Yeah, I think this is like really groundbreaking for AHC, of course, but it sounds like also for possible other neurological diseases that could also be affected by the work that's being done. So it's a really big deal.
And that's kind of exactly what we're hoping to do with Rare Hope. And I think, we're used to, in our AHC community, we're kind of used to this, having patients that are a cluster of different genes with a cluster of different mutations. And it's not just a black or white, this is AHC, this is not AHC. We're used to this kind of grouping, a genetic grouping. And that's just part of our disease community. And I think that is very much kind of the intent of Rare Hope as well. It's to look for these commonalities of either biology where there are genes that when they mutate, they produce similar sets of symptoms in a similar type of patient. So we already have that in AHC, but we're also looking for ways where we can apply a type of therapeutic across related genes. So where we can think about having clinical trials in the future, whether it's for something like gene editing or whether it's for a repurposed drug, where you could look across a set of patients that might have multiple different genes, multiple different mutations on those genes, and approach all of those patients together at the same time. And maybe it's incremental, and it's something that needs to be kind of navigated carefully and precisely, and it's something that I think hopefully that the FDA and regulators are increasingly open to, this kind of basket approach where there are multiple different patients with different profiles that can all be treated together more quickly. And that's kind of the key piece that we're really excited about with Rare Hope especially.
And hopefully that resonates within our disease too, because there's so many people that they've been clinically diagnosed with AHC, but not genetically. And even though they're showing exactly the same symptoms as our children, they are, not classified into a genotypic bucket. So, that is something that we're trying to address more widely than AHC, but certainly within AHC.
That's such an important thing. I'm really glad that you hit on that topic, and that's something that has affected our family too, because Dallas has multiple gene mutations, and they're all in these different sodium and potassium areas, and so I've never known, if a gene editing treatment happened, if it would really apply to us. So that's why I'm really excited about what you all are doing at Rare Hope, because of the fact that it could, affect and help so many others. There's lots of people in the community with all these different genes, and so I think knowing that there's hope and progress happening for them, even if not in like a very straightforward manner, that they're still being thought of, I think is really, really important.
And we're really, Annabelle has a rare variant. There are not that many patients with her variant. She's not one of the more common variants. So I think we've kind of always approached it as how can we how can we improve the lives of the greatest number of patients? Sure, let's advance research for the most common variants. That's an easy ask. The more complicated challenge, I think, is finding ways to approach some of the rare variants, thinking about ways to cluster patients using, say, different types of omics data, using ways that we can kind of look for maybe biomarkers in common with a cluster of patients that might suggest that those patients might be amenable to the same repurposed drug or the same type of therapy down the road. So I think we're looking for ways of looking for biomarkers that could be validated in a cluster of patients. We're trying to make sense of that kind of spectrum of different patients and find clusters that make sense that we could target in a way that might make things a little bit more accessible for patients with rare variants.
Yeah, and I want to kind of underline that point that Nina just made because I think it's so important. There are so often ways to kind of break communities apart, look for individualities within diseases. Like, okay, the A1N is very different for E815K, which is very different from L839P. There are, of course, ways to do that. And, say things like, well, that child doesn't have a genetic diagnosis. That's divisive, right? What Nina's talking about is looking for commonalities. Like, what are these things that are common across patients, and how do we cluster these in many, many different ways? And one example is genes. CACNA1A and SCN2A and AHC, ATP1A3 and ATP1A2, they all have patients that look exactly the same, right? How do we figure out why? And how do we use that to our advantage?
I just love the heart that you both operate from, because I think the fact that you are parents to a child with this disorder and you're connected and close with the community, it really, I think, informs so much of what you do, because like Simon was talking about with the uncommon mutations or the people who have no known gene or a very unusual gene affected, or older patients and people who can kind of get forgotten with all of the excitement around new treatments. I love that you are not forgetting about them, that you're still trying to keep an eye on like what could help everybody, and I know you've talked about, like, I believe your word for it was like a platform approach, of like looking into all the different avenues that are possible. Could you expand a little bit more on that and kind of explain to us like what your role is, Nina, and with Rare Hope, and like how the structure of research for AHC actually works in like real time? Like what those meetings are like, or I think it would just be really interesting to know kind of how that is occurring behind the scenes.
Sure. Yeah. The sausage making, they say. Well, it's, I think it's funny. I come from an odd, management consulting background, so I kind of like to have frameworks and organize things. What I should say is my actual background, which is my expertise in masculinity in 17th century Bermuda, which I always like to get a little like shout out to my actual academic training. But the consulting background, I think, has been very useful in that I've liked to think about things, and Simon too. I think we both, from his kind of business background, I think we like to think about the big picture and how we can lay the best possible path to achieving our objectives. And I think we early on determined that this would be multiple paths forward. So I think that, there's the big picture of kind of looking at gaps and looking at the different paths forward and going down as many paths as possible. But I think we've also really kind of changed the paradigm of what individual projects look like. We might identify a gap, identify the right team, talk for several months about how best to structure a set of experiments that can help us answer a question in the most direct, cost-effective, time-effective, efficient way possible. I guess efficient is a better word than time-effective, but anyway. So, I think we are very, and this is with Natalia's input as well. I think Natalia really trained us to be very, very interested in the experimental detail, because there are, in any of these projects, it's not just kind of the construct and the initial design of what a project is going to look like. It is the constant hundreds of decisions that need to be made along the way. And I think the way we structure things is that we have enough kind of back and forth with researchers that we can tell when there's a decision point. We can understand the different kind of factors to consider, and we can kind of help weigh in on what the best path forward is. And I think everyone really benefits from that. I think we are so deep in the science that it's not an odd thing for us anymore. I think we have enough kind of subject matter expertise generally that we can get to weigh in on decisions in a way that is hopefully constructive. And I think it's something that, researchers, it's not usually just one researcher and us, it's usually a team of lots of people with opinions. And so kind of coming to consensus, and we are, inserting that patient perspective in the larger consensus that's happening. I think that's something that's also important, thinking about how to navigate. You have the plan, you've thought through the plan really, really carefully, but you're still navigating that, and you still have this map in hand, and there are things that happen that are not expected all the time. When you get through experiments and you find this, well, what do you do next, right? And I think having that exchange between researchers where we can be involved very close to the details in a way that's, we're not making people write reports all the time. We're just having conversations about, what's happening, what the best approach is, what are some alternatives, and then we're kind of helping make those decisions.
I'd like to explain maybe quickly from a kind of big picture perspective what I see Nina and Natalia doing a lot. When you look at other rare diseases, especially similar ones to AHC that are small, there are a lot of people working on these diseases, but they're working on them in silos. They are, for instance, looking at the molecular composition of a particular protein, or like using X-ray crystallography or high magnification understanding of what that protein looks like, for instance. But they're not looking at the big picture of it. Like, okay, how does that connect to network effects, to therapeutic effects, to mouse models and cell lines, and all of these different things that are informative to the bigger picture? Bringing them together is acting as a hub to all of these silos and creating a hub and spokes model where everybody is contributing to a central understanding. That doesn't often happen. And that's why, having somebody at the center of this so often works. Like many of the diseases that have therapies now that are rare are led by somebody who's essentially acting as that hub. And having a real understanding of the disease to be able to connect these dots in a strategic and a tactical way just adds so much to the disease. And one good example of that is how early foundations start. They essentially have to accept proposals from scientists, right? A scientist comes and says, I think this is a really good idea. And the foundation says, I don't know any better, so I'm going to fund you. That doesn't happen anymore, right? When somebody comes to Natalia and Nina, they say, guys, I think this is a really good idea. Nina and Natalia say, wait a minute, like, let's go through this in like excruciating detail and figure that out. Like, here's what we know, here's what you know, and here's the protocol. Like, let's get this to an A-team level, because otherwise we're going to say no. And so, I think everybody is making each other better in that way.
The other thing to point out is that, this is quite a time-consuming process. I think from Simon kind of digging in 2017 and reading papers and understanding every word and every concept in a paper, and it taking 20 hours to get through a single paper, but understanding the depth of knowledge and the details were important. I think that's the same kind of approach that we've applied throughout all of this process. It's been this constant learning process, but it has been something that is only possible with like a huge amount of time and energy expended on it, and it's something that we take really seriously.
So in just being curious about how the developments happen and how the research happens, what would be the process of research development and how does that work?
I think we always start, we have this baseline understanding of what patients are experiencing and what patients and their families care about. And I think that's kind of the foundation for everything. But I think, the next step is for us to think about the areas where we may have gaps and where we may need to fill gaps, where there's either a critical knowledge gap that needs to be addressed, or whether there's a new technology that might, lend itself to a therapy that might be useful for patients. So it's, identifying gaps. And then we really flesh out what a new concept would look like. We read a lot of papers. We think about how it might specifically apply to AHC. And then we look for who the best possible collaborators, the best scientists in that field might be. And we think of, the hardest possible questions to ask them. We will sometimes we work with a really great group of AHC experts. Sometimes we kind of bring them in, particularly if there's a really good reason to bring in a certain person because their line of work is closely related to whatever new thing is that we're exploring. And then we kind of circle up with them, think about this issue from their perspective. Often they bring some kind of concerns or some new questions. And so then we kind of implement that feedback in another loop and look for more literature that kind of answers the newly expanded version of what we've been considering. And so then we kind of keep refining, and then we think about who the team is that we should bring in. So then we talk to that team. We kind of go through that whole process with the team, with those kind of field leaders that we've identified as being like the people to work with. We ask them lots and lots of questions. We bring in our scientific advisors, we get their input, we kind of introduce the new team to the existing group of AHC researchers that we're already collaborating with, because that's often this very kind of interesting bidirectional exchange where there's useful questions and comments on both sides. And then we kind of keep refining the concept. And then once we know what we really are interested in, then we'll ask for a proposal. And we usually put some pretty tight parameters around what that proposal should look like, whether it's, which milestones, what questions we want to answer. We put some real kind of guardrails around, like we want the most direct path that is still kind of rigorous and, meets kind of our pretty strict criteria for, reproducibility and things like that. So we ask for a proposal, then we think through the proposal. Once we receive it, we kind of iterate back and forth a number of times. And then we, Natalia and I go back and forth typically, 3, 4, we go back and forth a number of times. We try not to be too annoying about things, and we try to suggest very clear changes if we do want to request anything. And then we usually bring the proposals to a council that is a group across the different AHC foundations, and that gives people the chance to kind of weigh in and ask questions, express concerns. Sometimes we bring the researchers back in again. We are always kind of iterating, revising, figuring out how best to incorporate everybody's concerns and background and different questions. And then we kind of finalize a product with kind of input from all of these directions. We come to a consensus in that research council group that this is something that we want to recommend, and then we bring it to the different foundations and ask for funding support. And that's just kind of the first piece, because then the next piece becomes the project management, the project oversight. Management, in some cases, oversight in others. There's some places where we're very, very intimately involved with the day-to-day decisions. And then there are other places where, we just want monthly meetings. We want to be apprised of things, and we want to be able to weigh in as necessary on different decision points. But that's kind of what that process looks like. Sorry, that was a long answer.
Okay, so Simon, for families that are hearing these terms for the first time, such as gene therapy, gene editing, ASO treatments, what is the difference between these therapies, and what kind of insights can you give us in layman's terms?
Sure. So I'll start just by giving a very quick description from a scientific perspective, and then I'll give analogies on how to think about this. But gene therapy is essentially delivering a functional copy of a gene to as many cells as we can get it to, so that that functional copy can make functional proteins. Gene editing is changing the DNA of in as many cells as possible, so that again that DNA can make a functional protein. And then antisense oligonucleotides, or ASOs, are blocking the ability to form mutated proteins. So those are the three different things, and I'll give you analogies on how these things work. So if you think about your DNA as an instruction manual on how to create you, and each page or each chapter is a gene that explains exactly how to form a particular protein that makes you up. And in your case, there's something like 22,000 different chapters that comprise you. So it's a pretty long instruction manual. But what we're trying to do with AAV-mediated gene therapy, ASOs, and gene editing is to change the book that describes a particular protein that's being formed. And that protein is called the sodium-potassium pump with most people with AHC. And that instruction manual to make that particular pump has some errors in it. So the errors are causing that particular protein, and you can think about it as a little molecular machine. It's causing that molecular machine not to work properly. And in some cases, it's making a machine that's hurting you instead of helping you. So we want to change the instruction manual so that the instruction manual is either correct or it's not producing that machine that's hurting you. And that's what these three different therapies are all about. So in the case of gene editing, we're going into the book and we're cutting out words that are wrong, and we're pasting in words that are right, so that the instruction manual works properly again. Right? It's a very precise instruction on how to create that protein, that machinery. You can get even more advanced than that with gene editing, and you can use whiteout instead of cutting and pasting. You could just white out and write over the top of it without damaging the book in any way. And that's the most sophisticated version of gene editing that we're trying to use at the moment, and we have actually rescued a mouse model with. Now, if you think about AAV-mediated gene therapy, it's as if we're taking a whole chapter and we're inserting it into the book. We're not removing the chapter that was wrong, but we're inserting a chapter that's right. So now you've got two chapters, one that's wrong, one that's right, but you can refer to the one that's right if you need to, right? You're diluting the effect of the one that's wrong. That's what AAV-mediated gene therapy does. It delivers the right message. And then ASOs, if you think about ASOs, it's kind of like taking Velcro and pasting the Velcro over the top of the part that's wrong. So that not only can't you see that instruction, but you're going to stick those pages together so that you can't read those pages at all, so that you can't produce that bad machinery. That's kind of like what ASOs are. So these three different methods can be complementary. You can change the DNA within the book. You can stop it from being able to be read, or you can provide a whole new functional chapter. And you can do all three if you want to, in a single patient, which is difficult to do, but possible to layer.
Wow. That's really good analogies. I love Simon's analogies. There was one speech he did that was like, I don't know, hippos in ballerina tutus and like cats as witches, and I don't know, he always has so many fun analogies. So I was waiting to see what you were going to come up with today.
Hopefully that's a little more explanatory.
Yeah, I think that was really helpful.
Great. And now just kind of to relate some of those terms, how does this apply to what people are hearing about with CRISPR, and why is that such a big deal? Kind of like you had briefly mentioned that the mouse model had been rescued, and kind of connecting those pieces together.
Yeah, so I'm going to use basically three terms in my answer here. So the first is CRISPR, right, which I think everybody has seen and is aware of in the media. There was a Nobel Prize that was just handed out recently on the folks that created CRISPR, Doudna and Charpentier. And that was the first form of gene editing that was actually usable. So that's a somewhat primitive version today, right? That's the cutting and pasting that I was talking about. It damages the book. But there are other rounds of that. So the next round was base editing, which does not cut and paste in the same way. It's not as damaging. And then the most recent round is called prime editing, which is, that's the whiteout version where you're not damaging anything at all. You're just, pasting over the top of it. So it's we use prime editing, the most sophisticated current version of gene editing, to rescue a mouse model with the AHC, and it worked better than we could have expected. It felt like we were doing something sci-fi, but that sci-fi work that we did actually did work, and we were able to rescue our disease, which obviously is a big step towards clinical trials and therapies in humans.
And am I right in thinking that this is one of the first of its kind in terms of a neurological disorder or nervous system disorder?
Yeah. So when you've got these three different types of CRISPR, you've got the three different sophistication levels. For this sophistication level, for the prime editing version, which is the most sophisticated, it's the first time that a neurological disease has been rescued in vivo, which means in an organism, for a neurological disease. And we did it in a mammalian model, so a mouse model, and we're able to rescue those mice. So the mice essentially had their disease significantly mitigated. Not cured, because you can't get to every cell to correct every cell in the body, but the disease was mitigated enough that we could call it a rescue.
I don't know if I realized that's what in vivo meant. That's like really exciting to realize. So that's the first time in a mammal that this has ever happened.
That's right. The first time in a mammal to rescue a neurological disease, which is huge for us, because trying to edit cells in the brain is a really hard thing to do, and we did it with enough efficiency and effectiveness to be able to rescue a mammal. That's, for me, it doesn't sound like it's possible, but we did it.
It's amazing. And so other treatments that have been successful and kind of come before this, were those in like different body systems than the brain, like a kidney disorder or a type of cancer, things like that?
Yeah, so the way I think about it is that we've been able to correct diseases using gene editing, starting with the simplest form. And that was basically ex vivo. And by ex vivo, it means outside of the body. And so for things like sickle cell anemia, you're able to take the blood out of the body, correct it, and then put it back in, right? So that you are doing the work without having to do it in a living organism, even though you're doing it with living cells. So that was the first step. So they did that. And then you start targeting systems within the body that are easier to target than the brain, for instance, right? Everything goes basically through the kidneys and liver, for instance. So targeting those. And now we're getting onto the most sophisticated structures that are hardest to get at, like the brain.
So follow-up question for that, and this is a question from the community. Is anyone actively working on gene therapy, gene editing, or ASO-based treatments specifically for AHC right now? And I think I know the answer, but I would like to hear what you have to say.
So yeah, the short answer is yes. The longer answer is there's an enormous number of people working on this right now. And it's not just kind of direct work, it's also indirect work. So if you imagine you've got to develop therapies, right? And that's all being done. And it's being done by a lot of people. We've got these multiple different therapies that are being developed at the same time. But you also have to understand the mechanisms behind it so that you can do both in tandem, in parallel. And we've got a lot of people working on those mechanisms too, so we can understand both while we're producing these therapies to see if they work. And one is not holding up the other. We are making sure that as we test to see if things are working, we're then backing that up with mechanistic science, and vice versa. The mechanistic science is informing the therapy development. So it's nice to be able to know how the disease is caused. And there are different ways that that can happen within a system like the brain. And the brain is made up of so many different neurons that it could originate in part of the brain, or it could be a whole brain effect, or it could be some type of mechanism that causes a spreading issue, like spreading depolarization, or epilepsy, like seizures. And we want to understand how that all works, not just at the network level, but at the cellular level, so that we can see, what proteins are doing to cause that whole chain of events that ends up showing up as AHC. So when we know that, it helps us understand, okay, like here's where things are breaking, here's how things are spreading. How do we stop the breaking, and how do we stop the spreading? And that's how we're developing therapies, because it gives us targets.
And where are some of the places that those therapies are being developed, if it's okay to share?
Yeah, sure. So one of the basic philosophies that I think the whole team has embraced is that we really only want to work with the best in the business. So we've been really targeting people that are essentially at this point household names, but people that really understand this disease and really understand the silo of work that they're doing, that can contribute to the understanding of the disease as a whole and the development of therapies. So these are just examples of the people we're working with, but everybody's heard of Harvard and MIT and Northwestern and the NIH. We have labs at all of these locations. The Broad Institute is part of Harvard and MIT. Some of the world's leaders that I think should be winning Nobel Prizes are at these institutions working for us. The NIH, we have a dedicated lab to sodium potassium pumps and essentially AHC and our disease with some of the best minds in the business. Northwestern, we have a dedicated lab over there that's looking at diseases like ours and diseases that are similar to ours to look for interventions. We have the Children's Hospital Network around the United States, Children's National, Boston Children's, that are all behind us on this. And the therapy development is not just one team, it's people across all of these institutions. We've got we've got people that have been doing this for 30, 40 years that understand this pump down to the atomic level and can predict changes that in humans, like what type of symptoms you might expect by changing one atomic molecule. So it's about as sophisticated as it gets across people that have multiple different expertise types that are contributing to a huge combined effect that the whole is significantly greater than the sum of the parts.
So in any of these treatments that we're discussing today, if they were to be successful, we know we've had some successes in mouse models, if it was translated into a treatment for humans, and I know this is all hypothetical, but what would we expect to see reduced? Would we expect to see reduction of episodes, reduction of the kind of baseline issues like gross motor, fine motor skills, speech? Does it depend on the age of the patient? Kind of what kinds of things would we expect to see, and what considerations would we need to also have in mind?
Yeah, so that is a very tough question for a layman to answer. I'm going to do my best here, but obviously massive caveats around me not being a neurologist, etc. So when what we're trying to do ultimately is to cure this disease. Which means elimination of symptoms. But that is at this point more of a dream than reality. It's very, very difficult to cure a disease like this across every cell in the human body. What's more likely is that we can mitigate the disease. And in order to mitigate the disease, we're prioritizing. So, what are the parts of the brain that we should prioritize as the most important? Like, where does this originate? Can we stop these episodes from happening? If we do stop them from happening, what types of symptoms do we correct? And then that starts becoming a very difficult question, right? And to your point, like, does age matter? And if you're not curing someone, then yeah, age is going to matter. Like, we have synaptic plasticity, which is essentially like how the brain is developing over time, right? That's a child's brain has had none of this development. These neuronal networks have not all kind of connected for them yet. So it's easier to change the behavior of the brain in an infant than it is to change the behavior of a brain that's fully developed with all of these neuronal connections. So if you have a patient who has a disease in a particular part of the brain, or like neurons that keep failing, for instance, that brain is likely to develop around that problem. So instead of connecting neurons at a point that's going to fail, it connects at points that are not going to fail and develops a network very differently from how your or my brain might develop. So what will happen if we provide one of these therapies in that case, right? And what we've been working on as an assumption is that the earlier the better. The younger the patient, the more likely that we're going to be able to correct longer term symptoms. But that doesn't mean that we can't prevent episodes. It doesn't mean that we can't reduce severity. It doesn't mean that we can't reduce the length of episodes. And all of those things are positives for our patients. So it's a long way of saying we don't know exactly what's going to happen, but we're trying to prevent all of these things.
If an AHC patient has epilepsy, is that become more complicated to correct? Is that put a different risk on it? Is that one of the main symptoms that you're looking to focus because of the risks behind an epileptic seizure? How does that change the research?
So from the papers that I've read, patients that have epilepsy tend to have more severe symptoms. Now that is just statistically speaking, right? That might be very different from patient to patient. But epilepsy and epileptic seizures gives us a good target for measuring whether or not a therapy is effective. So even though a patient might have epilepsy, it might actually be helpful to have that patient in a trial because it's easy to measure, like have those epileptic attacks become less severe? Have they stopped? Are there fewer? That's an easy measurement and a good way to identify whether or not a therapy is working before or after or during a clinical trial. So it seems to be more severe, but it also seems to be more measurable.
So would the epilepsy also be improved / cured with the same treatment that would also treat the episodes in a hypothetical situation?
It's impossible for me to answer, but obviously that's something that we are looking to try to achieve.
But it wouldn't be, I guess what I'm asking is it wouldn't be a separate treatment because the gene change is the issue. So the hope would be that it could solve a wide variety of symptoms. Is that accurate?
Yeah, we're trying to reduce or eliminate every type of episode, whether it's dystonia or hemiplegia or epileptic attacks. It's we're trying to reduce all of them, but it's hard to know what's going to work and what isn't. We may see a result where certain symptoms are reduced, but others are not.
So, realistically, with the recent in vivo prime gene editing success, what is the next step past that? What happens next with that specific study? Are we close to human clinical trials at all? And what do we have to look forward to in terms of next steps?
Yeah, that's a great question. There's a typical timeline for the development of a therapy, and that therapy has to go through multiple steps. So we've been through very challenging steps up to this point already, which is really good news for this community. In order to get a therapy to this point, and I call this point proof of concept, right? It's the point where we know that this works in a mammal. Now, can we just make it work in a bigger mammal? Is the question. And but the steps we've already gone through are to develop all sorts of models, and we call these things models because they model the human condition, but at a much smaller level, a much more precise level. So, for instance, a cell can be a model of a disease, right? A mouse can be a model of a human disease. And the more sophisticated you get in terms of the number of cells and the bigger the animal, the closer we get to being able to replicate the human condition, essentially. We've been through lots of those stages. So we've developed the therapy, we've injected it into mice, it works in mice. Now we need to make sure it's not toxic in humans. So that's most often done in a rat model, but sometimes it's done in a monkey model, a non-human primate. Then we need to make sure that the FDA agrees that this therapy is ready to go in humans, and their whole job is to make sure that it's safe, which is very difficult to do when it's a brand new technique, right? There's no precedent here. We've got no other disease we can point to and say, it worked in the brain in this disease. We're doing something very similar for our disease. You should approve it. We have to, argue the theoretical case with the FDA and say, here's why we think it'll work. The FDA says, have you considered X? And we say, yes, we've considered that. Here's why we think it'll work, why we think the risk is mitigated. But if they say, have you considered Y? And we say, actually, no, we haven't considered that. Then we need to go back and figure it out and come back to them and say, here's what we figured out. What do you think? And then it becomes a negotiation, right, between how much does that patient and this patient community need this? Are they going to die if they don't get it, basically? And okay, this patient population is not massively at risk, but you're introducing risk. So, is that risk warranted? So that's the push and pull between us and the FDA at that point. Once they approve it, then there's a clinical trial. You need to design that clinical trial really carefully, and we've got top people in the business that are doing that, like Uncommon Cures and Marshall Summer and his team. But once that's well designed, and that design includes how many patients, what genotypes, what ages, what are we looking for exactly? Are we looking for a reduction in episodes? Are we looking for a reduction in severity? Are we looking for specific symptoms to change? Then we enroll patients, we get them into the trial. That's going to be at a hospital or a decentralized clinic, and then we inject people. And then we see what happens, right? So it's like you're injecting mice here. It's an experiment in humans. And we see what happens. And then if we can quantify therapeutic effect adequately, then the FDA approves it, and we start manufacturing it for the entire patient population. And then pharma starts to sell that as a FDA-approved therapy. Those are the steps, and we're at proof of concept before toxicology, before clinical trial recruitment, before the FDA negotiation. So all of that needs to happen.
Kind of to follow up with that, realistically, since there are people working on this, and now we have seen that this first mice model was rescued, what is the timeline moving forward, and what realistically can people anticipate?
Yeah, so as you can imagine, that's quite a hard question to answer. I'll do my best. We have had cases like with Millicent, which is an ASO. Julia Vidarello developed that for her daughter Mila. She did that in record time. So that was something like 1 to 2 years from development to clinical trial in Mila. That is the fastest we could possibly expect, right? So that's one bookend. On the other side, there are therapies that go for many years in FDA negotiations before they get to the point where the FDA will allow it, and sometimes the FDA does not allow it. So in the case of ASOs, and this is, I'm talking about Millicent again, ASOs had been used. It's not a new therapy. It has precedent, going through the FDA, and also has, pharma is developing this for multiple diseases like spinal muscular atrophy and so on. There's an established path for that, so it could be quick. Gene editing, especially prime editing, does not have that same path, so it can't possibly be 1 to 2 years. So the best possible guess is like 5 years at this point, and even that is somewhat optimistic.
And what was the disorder that was treated with that ASO you were discussing?
What was Millicent?
Batten's disease, a rare form of Batten's.
Something else that brings to mind is just how groundbreaking this has been. It sounds like this has really kind of made AHC more well known within the scientific community and perhaps the circles that you all are running in. Nina, would you be willing to share the story about the previous NIH director and kind of what happened there?
I would love to. No, I think we should all be so proud of the position that AHC now has kind of on the map of rare diseases, because we are in a position that, we never were before. And so I think, I think of the day. So I was with a friend right before Christmas, and we went to an event at a church in DC, and there was Francis Collins standing there chatting to someone, and I walked into the room and was like, oh, that's Francis Collins. Okay, this is, Francis Collins, the former head of the NIH, who, started the Human Genome Project, who is, one of the most illustrious scientists of our day. He's here. I'm going to go talk to him. And so I make a beeline for him, and luckily my friend wanted to talk to the person he was talking to, so we kind of had this perfect interrupt the conversation and derail it for our own purposes moment, which it's actually rarely worked as well as it did in this one. But so I go up and I say, I'm just so delighted to meet you, and I introduce myself and I say, my daughter has alternating hemiplegia of childhood. And he says, well, there's some really good work being done on alternating hemiplegia of childhood right now. And I said, well, yes, there is. Thank you very much. Because he knew about our disease. He knew about the research program that we built for AHC. And, from the moment we met, we ended up having, a long conversation. We've emailed multiple times. He's a wonderful guy. And, one of the people in the world that I truly respect the most. And he was familiar with our work. I mean, it's a remarkable thing. It was really something.
Yeah, especially if you think about how the fact that our patients are considered 1 in a million, that there's I think I read a statistic this summer that was there's probably 1,500 or less. And thinking about that in terms of my high school graduating class, I was like, oh, wow, that really puts into perspective how few of us there are and how truly rare this is. But then at the same time, this is the disease that is having this massive breakthrough that's never happened in a mammal before. And so that's just really incredible to think about. This teeny tiny community is just a powerhouse, truly. And I know it's been a, collaborative, and there's so many organizations involved and so many people funding and so many researchers, but ultimately this is a very small disease. And so how phenomenal is that?
It's pretty phenomenal.
Oh, that's awesome. Thank you. That's really helpful to know because I think there's so many organizations involved in AHC, and I think it can be difficult to kind of know like who to contact, who's spearheading what, and if there's a research initiative going out, who is kind of taking the reins. And so I think that kind of helps flesh out how that all works.
Well, I profoundly think that as a community that communicates with one another, that kind of doesn't operate in our own silos, that we can just achieve so much more. The idea of duplicating work or paying for something twice, or not being able to collaborate across different entities, it would be such a shame. So I think we've had a really good way of collaborating and jointly funding, jointly sponsoring a lot of these exciting projects as they come up.
So there are plenty of examples. We have certain proposals that have come in that are NIH quality, and I'm thinking right now about like Kathleen Swetner's proposals. They're long, they're detailed, they're very explanatory. They are they're from an expert who has 40 years of experience, distilled down into a level where novices can understand it and experts can understand it, and there's nothing in there it's simplified, but it's not simplistic. So it's enough detail to really understand what we're going to do with a real milestone-based approach that's backed by the best quality of science. And if something is not of that standard, then we push back until it is. And that's the set of rounds that Natalia and Nina go through before the foundations ever see anything. So by the time they do see it, it's sophisticated, but at the same time simple and easy to understand, so that we're making decisions based on details, but also on a simplified version that stands up according to structure.
Yeah, that's really helpful. And in thinking about the data that's often collected in these studies, I know we had discussed a little bit about how that's something that you guys are still interested in, is like biomarkers. And is that something that is going to be happening at the family meeting or at any point in the future that we could all, as patients and families, get involved in?
I think patients can be so involved in research, and really should be, and whether that is participating in any of the survey platforms, which is a really great way to kind of register your family's experience with the disease, and to kind of make your preferences for which symptoms we should really try to target with a therapy. That's one thing. Surveys about triggers and sleep and other kind of topics of interest both to researchers and to patient family members for managing the disease. I think that's a really great way to insert your child's experience into what we are advancing for AHC research. The other, the different, we have a wearable study with Anupam Gupta. I think the MGH team that Dr. Gupta runs was just so impressed with how quickly we could enroll the number of patients to fill that study. I think they imagined that enrollment would take a certain number of months, but it was a certain number of like weeks or days, because we have such a wonderfully motivated patient population. Because data about your patient, whether it is the wearable data that we were collecting on submovement differences in the AHC population, that's critical data to understanding the disease, potentially flagging events before they happen, and ideally, marking AHC type submovements with differentiating those from the wider pediatric population. And so by contributing your patient's data, whether it's samples, blood samples that are also incredibly valuable, or these kind of experience-based biomarkers like wearable studies, I think these are ways that are just so profoundly valuable to the researchers that we work with, but they're also kind of centrally patient-owned resources that are really incredibly valuable for shaping the direction of research.
And I want to underline that one too. There are so many critical areas that patients can start to be to really drive the process here. One thing that I would say is most important is getting in contact, like having contact details available, getting in touch with Izzy if you haven't yet. Make sure that we can access you and vice versa very, very quickly, because when therapies are developed and we're qualifying people, the first thing we need is contact details. The second is genotyping. If you don't understand the genotype of your child or your patient, then make sure you do, and we can help you to do that. Understanding which gene is affected, what particular mutation you have, again, is very critical. And then the biosamples that Nina mentioned, blood, that can be translated into cells that can be used to test whether a therapy works in your child or not. That's also quite important. So we've talked about it a little bit before. If you can do these things, including getting involved in Nina's surveys, then we really we're putting your child ahead in terms of the ability to get them treated.
And I should say too about the survey platform. I mean, this is an independent review board. This is a survey platform that is protected. The data is protected. The data is only shared if it's de-identified. It is a formally conceived database and registry and survey platform. This is not just kind of like a casual take a survey and your patient's information is going to be like out there in the world. It is a very protected data set that we take very seriously, and we work with an independent IRB to make sure that all of the kind of safeguards for patient data are respected, and so we take that responsibility of housing that patient data very seriously.
Yeah, and as an AHC mom, I think it makes me excited knowing that when you have that data, that's helping to inform the research that you're looking at and also the treatments. And so I think that is really valuable to know that getting involved in the surveys and the research, it's not just helping the research, it could be helping your child in the long run.
And I think that's a good point too, because we have a registry as well. So if you don't want to take the full survey, there are 2 surveys, and I think together they probably each take 15 minutes or so. So there is, there's some time commitment there. But if you just want to be in the registry and provide some of your demographic information and contact information, that's also really valuable to us. So that's something that's useful as well.
Okay, so kind of segueing from that, those physical components, participating in the surveys, the wearable study, things like that parents can be contributing. Obviously, all of this comes with a financial responsibility. And how can parents best be advocating for their kids? Is that fundraising? Is that contributing to what is the best, just contributing to the efforts that are already happening, or specifically for their children? How financially does each family need to raise $1 to $2 million for their kid?
Yeah, and I can take the first crack at this, Nia. It would be great if you jumped in on this one too. But right now, for me, that is not necessarily the highest priority, raising money for your child's ultimate treatment with one of these big therapies like ASOs, gene therapies, or gene editing. The most important thing is to make sure that you are in contact. We have all of the tools available that we need to be able to qualify your child for a potential clinical trial. The major priority right now is to make sure that these therapies work, because there are different levels of contribution towards the cost of these different therapies that are going to help your particular patient, whether it's through clinical trials contributions, or later on through things like Medicaid, Medicare, or health insurance that may be able to contribute towards the cost. Right now, there is a cost involved in developing a therapy. There's a cost involved in having a clinical trial. All of that is pre-marketing of an FDA-approved therapy. And traditionally, those are paid for by the research institutions, by the manufacturer that's going to be profiting from this ultimately down the line. and from foundations that contribute towards it. So, for the particular patient that is involved in a clinical trial, often there isn't a huge financial burden to them or their families. So those patients are essentially getting treated for a very low cost. Obviously, there's a risk involved in a clinical trial that there isn't post FDA approval. Once it is approved by the FDA, though, there are also avenues to raise capital for your particular therapy. We don't know what those therapies are necessarily going to cost today. $2 million is a good guess because we've seen that in diseases like spinal muscular atrophy. But that doesn't mean that you're coming out of pocket for $2 million. This is going to be a community effort to raise capital for our kids. If a particular therapy works, then it's all hands on deck to make sure that we are treating as many possible patients. It's not about what you can do, it's about what we can do to treat your child. And we means the government, through Medicaid, Medicare. It's your payer who we'll be negotiating with as directly as we can to make sure that your child is covered for that particular life-altering therapy. And then it's about raising money as a community, foundations, to be able to, help supplement what this is going to cost. So it's, and then making sure as many patients as possible can qualify for a clinical trial to the extent that they need it medically or to the extent that they need it financially, or can benefit significantly. So these are all considerations. But for today's purposes, I would put, the 2 million price tag as a lower priority than let's make sure this thing works.
I think that's really helpful to know for families who either haven't started fundraising or who can't fundraise for whatever reason, don't have the right connections to be able to do so. It's just good to know that the money isn't going to leave people in the dust.
I think it is our expectation that families will not need to fundraise for some of these therapies and that there are other paths, that there are other ways that patients can be treated. And, what this means is that it means that Simon and I and others in the patient foundations, we are not the arbiters of who is enrolled in a clinical trial. There are considerations based on, patient's profile and their risk benefit and various other factors that we have absolutely no control over whatsoever. So I think, it is something we are anticipating that, patients will be enrolled in clinical trials in the near-ish future, right, in the next, say, 3 to 5 years. But that there is no fundraising necessary to be in that position. I think, the thing to say is that these are really big numbers, and they are not numbers that are necessary targets for our patient population. And I think that's really, really important to say. The other thing to point out is that there have been a few family members who have wanted to, make a contribution in a significant way. And I think where those have been the most successful is where they have also kind of provided a way to benefit the entire community by being kind of completely transparent, by, involving me or involving others in the community, so that what they are doing is they're not creating a new silo, they are advancing research that benefits everyone. And, Mary Saladino has done that really wonderfully. Christy Roy's done that really well as well. So there are ways in which people are, able to raise some money, thoughtfully deploying it, but by making sure that they're not just kind of hiding behind a little silo and doing something to benefit one person. I think they can go further, and I think all of us can benefit.
The beautiful thing about those examples is that those folks are working together. So, they're not doing something on their own. They're making decisions based on, how can we slot this into a bigger strategic plan so that this does benefit everybody.
Leading into that, and obviously, like you said, there realistically a goal would be 5 to 6 years, which honestly is incredible that it could remotely be that close. But with that being said, obviously in rare disease, time is of the essence. What does that mean for families? I know, obviously, gene therapies, gene editing is talked about widely across a lot of rare diseases. And I know specifically there's one mom who openly talks a lot about the Sanfilippo, and kind of unfortunately her daughter specifically is 5 years old, and she's gotten to a point in her Sanfilippo disease that even if a therapy was made tomorrow, it wouldn't be of benefit to her daughter. Do you find in the AHC community that there is a harsh deadline, that there is kind of, is there optimism in a later term cure or therapy, and kind of what does that look like for parents as their kids are aging?
I think we are so acutely sensitive to that ourselves. I mean, Annabelle is 10. So this has been something from when Annabelle was 2 to Annabelle is 10, right? This has been something that has been a question for us as well. So we address this as parents, as well as, as folks who are trying to help kind of lead the research efforts. I will say that we don't necessarily know. I think that is not a reassuring answer, but I think those are the experiments that we're most interested in, in the coming months, and literally in the coming month or two, we will have some insight. I don't know how much we'll be able to share, but we'll have some insight from our gene editing project on, what that therapeutic window looks like in mice. And that doesn't necessarily constrain, a therapy in patients. And I think from what I've heard of any future clinical plans, and some of those plans are more advanced than others, there is at this point no age cap for enrolling patients. And so I'm optimistic that, that the therapeutic window will not be something, it certainly I don't think would be as limited as it might be in some conditions. So I'm cautiously optimistic that there can be benefit to older patients, even with some of these treatments. ASOs have been used in 60-year-old patients. I mean, some of these, and it's disease specific. So just because an ASO is working in one condition in a much older patient, it doesn't necessarily mean anything either. But I just don't want to take them off the table until we know more. And experimentally, in our mouse models, that's kind of our best next step at evaluating that question. But frankly, we might go into a situation where we are treating a patient who is at the older end of what we think might be useful. And that may be the first time that we have a clear understanding of what the specific therapeutic might be able to achieve.
There's one thing that makes me really quite optimistic when it comes to AHC, though, and that is how episodic this disease is in nature. Like when you think about dystonia, you think about hemiplegia, you think about, choking attacks, you think about spreading depolarization, about epileptic seizures, nystagmus. These are all episodic. They come and they go. And, if we can prevent them from coming, then that's a huge advancement on its own. Even if it's not a cure, that prevention of episodes is still significantly therapeutic. So I think we've got some advantages when it comes to our disease, and at least something to be optimistic about.
I mean, that's such a good point. And that's why understanding what patients care about, and maybe even understanding what patients in different age cohorts care about, right? Like, what are the most daunting symptoms for patients, and can those be attenuated at certain ages in a mouse model, right? Like, these are questions that we are asking, and they're questions that we're asking with that context of like what patient families have told us. And so I think there are so many different symptom types in AHC, and there's so many different paroxysmal symptom types, but I think, to Simon's point, if we can mitigate some of those paroxysmal symptoms for patients, it will have a really big effect on quality of life. And so I also remain very optimistic. I speak conservatively, but I feel optimistic.
That's really interesting, because that's what I was thinking too, from like a non-science perspective, is like, I don't know. I'm not saying this is fact, but I would think that most likely we can't reverse, development. We can't go back in time and rebuild things that have been, taken away from a patient. But if we can stop episodes, that would be halting some level of disease progression that I would think would lead to a better outcome. Obviously, that's just my layman's understanding, but I think it's interesting to think about, halting the episodes could be really, really helpful.
we're trying to think across that spectrum. Like, how can we think about everyday life for patients, whether that's the hope for a cure or alleviating the most damaging or severe symptoms, or just making quality of life better. Those are things we think about all the time, and using a range or a portfolio of therapies to be able to achieve that is something we think about all the time.
Right now, kind of given where things stand with therapy and research and people picking up and the news getting out there and word getting out there about AHC specifically and the work that you all are doing, what brings you the most hope right now moving forward?
Sure. So, yeah, I mean, there's so many things that give me hope at this point. I feel like every one of these therapies is advancing significantly, like really fast. So we've talked about gene editing and how that felt like it was many years away, and now we've already rescued an animal model with gene editing. That's one example. We are seeing, to Nina's point, other different therapies and different technologies that are coming to market every day, like ways that we can bypass the immune system, for instance, to deliver multiple therapies, layer these therapies on top of each other. I think that's exciting for the future. I think the idea that every one of us in the world, like 8 billion people, could benefit from gene editing in some way, that's something that excites me. This might be something in the future that might be able to help not only rare disease patients, but everybody. And there's the ability to translate this and translate the work that we're doing into multiple other diseases. And then probably one of the bigger ones for me is that these types of therapies are, I think, the building blocks for an understanding of human biology. If we can see what doesn't work in humans and map that, then it gives us a real understanding of function and dysfunction in human beings. So all of those things make me excited about what this can do. Another thing from an economic perspective, we, just by kind of being at the sophisticated level that we're at and being at the cutting edge of science, we're getting a lot of people wanting to do things for us at very low cost to establish themselves and the process and the collaboration as something that's replicable and scalable. So we're getting a lot of advantages by being pioneers. That's from an economic perspective.
That is so true. I think we've gotten so many opportunities because of where we are now and how far we've come, and I think that's something that shouldn't be underestimated. So I think one of the things that gives me the most hope, we are at this time of immense technological innovation, whether it's things like gene editing, whether it's things like AI that gives us the ability to analyze massive amounts of data that could all of a sudden, become very, very useful. And I think over the next couple of years, it really will. I think that ability to analyze these data that can be, that, if you needed kind of human person hours to analyze the data, it's the task is just unsurmountable, but yet you are producing all this data all the time. And whether this is from our mouse models or whether it's from, omics data, from transcriptomics, proteomics, genomics data that we have as a resource, I think that the possibilities there are tremendous. But I think it's worth saying that the technology that is innovating, it has implications for us. That's not just these things like gene editing. It also means that we might be able to find a repurposed drug that could have an advantage for many, many of our patients, that it doesn't have to be this kind of high-tech therapy. It could be a high-tech approach to kind of a low-tech repurposed drug that could be accessible more quickly, that could be a benefit for so many more patients. And that's one of the things that really gives me hope. I think over the years, we've really tried to think about how can we maximize the value of what we're developing in terms of therapeutics for AHC to benefit every possible patient, whether they're a baby that's just been born, or maybe they're an older patient with really kind of severe symptoms that might not necessarily be a good candidate for a genetic therapy that's not going to be available for a few years, right? Like there is a spectrum of patient needs that we've been trying to address, and I think these innovations and technology that are happening in the background, and we're involved in some of them, right? Like we are doing some of the knowledge graph work with Elsevier, with other companies that are kind of helping us think through some of the repurposing work that we're trying to kick off now. We've started on that path, and I think as the technology advances and as algorithms become more mature and AI has kind of a cycle of validation, I think we will really be able to make some good progress that could be beneficial to so many patients. And it doesn't have to be this science fiction approach, which I believe in science fiction too. It's just, I think it's important to notice, as patients, that there are so many advantages of technology that, don't have to be this very kind of sci-fi technology.
So I guess to kind of put a bookend on this, if we think about, it's kind of been touched upon, but if we're thinking about families who are listening today, who are excited about the future, hopeful, don't exactly know what to expect, what are the best takeaways for them? It sounds like getting involved in the studies when possible, getting involved in the survey so that their voice and their patients' data is being considered when research is happening. Is there anything else that patients need? I guess making sure that they're in contact with us so that we can let them know. Is there anything else that you would want patients to know just around this whole topic of treatments and gene therapy and everything?
I think I've tried to convey our kind of interest in inserting patients into the research, but it's not just my perspective. I would say, and I would love for patients to know how much scientists that we work with and researchers that we work with, and obviously clinicians that we work with, patient experiences are incredibly important to all of these people. I think they are very interested in, AHC and AHC patients and understanding what they are enduring. And I think they're really committed and kind of personally committed to this disease in a way that's really touching and moving. And I would really like for patients to know just how, interested and kind of emotionally committed they are to this.
Kind of a quick story on that. At your Rare Hope Gala, we had the privilege of getting to meet so many wonderful researchers. And at one point, Tim Yu came over and introduced himself to us and was truly so in depth and wanting to know about Archie and what he was like, and was just so personable. But we unfortunately did not catch his name at the initial introduction and did not quite know who we were talking to. And we sat down to one of his team members and I was like, I'm so sorry, I missed his name. Can you tell me who that was? And he was like, are you crazy? I spent my whole life, I've moved across the world to work with that man. And truly, you would have had no idea. It was the most intentional, purposeful conversation. And so I can only imagine.
That's such a great story, and so kind of reflective of exactly what Nina was saying. Like this, if I were to urge everybody in this community to do one thing, that's get involved, because this community is so powerful because of the people that exist within it. And people like David Liu and Tim Yu and Kat Lutz and Marcus Terry and Alex Souza, and the list just goes, Kathy Swetner and Swoboda. I could just keep going all day long with like how wonderful these people are. It's and then the leaders of the community too, but every parent that I've ever met that's also is contributing significantly in their own way. Like every time you provide information about your child, whether it's symptoms or your experiences or your genotypes or tracking episodes on Stephen's excellent app, like this is a huge contribution to a global effort. And I couldn't urge you more to get involved in every way we've discussed.
And I think that's the perfect way to end today. Just thinking about there's so much power in our community. That's something that I think about and talk about all the time, is that I would say that of everything that I know about AHC in my roughly 7 years of being involved in this community, most of it, I would say 90% of it, has come from other parents and from the support groups and from being connected. And it's kind of amazing that we have such a very small community and there's so many powerhouses, so many contributors, and so many people just supporting each other. And when you're 1 in a million, I think that is kind of the only way through, is together. Well, thank you so much for being here today, Frost family. We have the highest respect for you and are so excited to continue to get to see what happens as we go down this road together.
Thank you, Zane and Henny. You guys are doing amazing work.
You are.
You guys.
I hope today's conversation left you feeling informed, encouraged, and genuinely hopeful about where AHC research is headed and about the real possibility of a cure within our children's lifetimes. You'll find links in the show notes for the key action steps from today's episode, including the Rare Hope Patient Registry, where you can register your contact details, the AHC research surveys, and the Ticking Time Bombs documentary by our incredible colleague Siggy Johannesson. Real hope is here, and we're working together for a better outcomes and a better future for our AHC patients around the world. Wherever you're listening from today, please know you are never alone on this journey. From our family to yours, we'll see you next time behind the episode. I hope this conversation left you feeling informed, encouraged, and genuinely hopeful about where AHC research is headed, and about the real possibility of a cure within our children's lifetimes. You'll find links in the show notes for the key action steps from today's episode, including the Rare Hope Patient Registry, where you can register your contact details, the AHC research surveys, and a link to the Ticking Time Bombs documentary by our incredible colleague Siggy Johannesson. Real hope is here, and we're working together for better outcomes and a better future for AHC patients around the world. Wherever you're listening from today, please know you are never alone in this journey. From our family to yours, we'll see you next time behind the episode.